Today, Metagenomics Lesson 2, we're going to talk about one of my favorite parts of, sorry, closing the chat window here. Metagenomics, which is Kmers. I called the first part of this lecture the power of Kmers, but what I really mean is the magic of Kmers, because they are quite magical when you get finding all the different ways that we can use them. So what we're going to do today is talk about the power of Kmers, how they're used in metagenomics, and talk about the first aspect of using Kmers, which will be taxonomic classification. So we'll start by talking about what taxonomic classifiers are out there, and then do a couple of demos using Kmers to do adapter trimming of raw reads, and then taxonomic classification of some metagenomic samples. So a Kmer is a word of DNA that is K bases long. That's a pretty straightforward definition. It's a very simple device once you get your mind around it. We can think about Kmers as different lengths and having different uses. So for nucleotides, we know that there's 4 distinct bases, A, T, C, and G. And so we can call those 1-mers, and there are 4 of them, or 4 to the 1-mers. At the level of 2-mers, we have AA, AT, AC, AG, da, da, da. So there's 16 of those, or 4 to the 2-mers. And it keeps going like that. So there are 4 to the 3-mers, and we also know those as codons. Here you have uracil in the place of thymidine for this table of the universal genetic code. And what I would say here is that all codons are 3-mers, but not all 3-mers are codons, because a codon is part of a coding region of a gene. So you could have intergenic regions where you can count up all the 3-mers you want, but those aren't technically codons. So it keeps going like that. Exponential growth, 4 to the power of x, where x is your length of your Kmer. You get up to 10 to the 19 unique Kmers by the time you get to a Kmer length of 32 using nucleotides. Amino acid Kmers grow much faster. They grow at a rate of 20 to the K. So for the same length of a sequence, there's much more potential diversity among amino acid sequences than nucleotide sequences. And that comes in handy later when we're talking about classification. So a lot of Kmers. What are these Kmers used for? That will depend on how long the Kmer is, and we'll talk about that in a minute. The first thing we have to do is, though, extract the Kmers from our sequence of interest. Usually these will be sequencing raw reads, or maybe contigs after you've assembled something. And Kmers are generally extracted by running a K-length window across all of the reads in your sequences. So for example, this read here, AGG, ATG, AGA, CAG, ATAG, is 16 bases long, and it can be broken into 11 Kmers of length 6. So the first one, AGG, ATG, we shift 1 base, and the next one is GGA, TGA, dot, 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 down the row. Another useful fact of Kmers is complementarity. So there is no privileged direction in terms of 5' to 3' that we can store the bases in, as long as we go the same direction. It doesn't matter what strand, sense or antisense, we store it in. So when the computer program that's doing the Kmer indexing or hashing is doing it, there's 2 potential ways to look at, for example, this sequence here. We have adenine A on the left top in blue, then we have cytosine red, then thymine purple, and guanine blue, going 5' to 3' on the left-hand strand. And on the right-hand strand, the reverse complement is CAGT. So we have ACTG or CAGT are both equivalent ways to store that 4-mer, that 4 base pair sequence. So the canonical way to do that is to store the lexicographically lower reverse complement. So in this case, A comes before C in the alphabet, so we would store ACTG and not CAGT. So if you look at a couple of other examples, if you had TTTT, you would store that in the database as AAA. TATA, the reverse complement is also TATA, so you would store that string, etc. Another fact regarding Kmers is that palindromes exist. So in molecular biology, a palindrome is a DNA sequence that is its own reverse complement. So you probably remember these from studying restriction enzymes, where the cut sites are palindromes. For example, this is EcoR1. It has a recognition site of 6 base pairs, GAA-TTC. The reverse complement of that is also GAA-TTC. And when the restriction enzyme cuts, it cuts and leaves overhangs that are reverse complements and identical palindromically. So a lot of tools, especially genome assemblers, require odd-length Kmers to avoid palindromes because these induce self-loops in de Bruijn graphs, which is the algorithm that is used for a lot of assemblers. So the reason for this is that an odd-length Kmer can never be its own reverse complement because the middle base always switches as you do the reverse complementation. So that you will notice when you're putting your parameters into various assemblers like Spade's assembler, that the Kmer length is always an odd number. So when we're thinking about the Kmer length, the length that you choose will be determined by the goal of your analysis. Not all Kmers are good for all analyses. So the most basic Kmer analysis you can do is essentially a GC content analysis where you take all of the 1-mers, that is G and C or A and T. Those can be stored lexicographically as A and C, simply. And that's used for all kinds of genome comparisons because different genomes have naturally small variations in their GC content. If you go up to 3-mers, you can talk about codon usage. For metagenomic binning, people generally use 4 or 5-mers. For taxonomic classification, people generally use 7 to 35-mers. And for genome assembly, usually in the range of 21 to 127-mers, you'll see. So the graph on the right here shows the number of Kmers for each K in an E. coli genome. So the yellow line here shows the universe of possible Kmers. That's if every Kmer was included. Whereas we're interested in the blue and the orange lines here showing the distinct and the unique Kmers that are actually found in the genome. So as you go, there's an increase in the number of Kmers observed. For example, if we're following the blue line, as we go to K of 10, 11, 12, 13, but eventually it saturates. And at that point, essentially every Kmer you observe in the genome is unique. And that's useful for genome assembly. So when you're using Kmers for any particular analysis, it's often beneficial to try out a few different Kmers and see which ones are best for your particular use case. I'm going to go through a couple of different use cases here just to talk about the history of Kmers and how they're used in various aspects of molecular biology and metagenomics. So the one point here is that the distribution of Kmers in genomes is not uniform. So even at the level of 1-mers, that is G plus C or A plus T, bacterial genomes have different but highly conserved proportions of these 1-mers. So this figure from a paper by Bolin et al. In 2017 shows how the G plus C content is distributed in the various aspects of the genome, the core versus the accessory, for a whole bunch of different bacteria. So what you can see is that, say you're picking one bacteria here, like H. pylori, which is in this light green. If you look at these box plots, you'll see that for each dot here, which is a region of the genome, the variance in the GC content is very low. These are all horizontal lines, essentially. Some of them are a little messier than others, especially in the accessory side of things. But in general, the variability is very low. And that's because across the entire genome, there's a highly consistent G plus C content among the genes and even in the intergenic regions between the genes. And that can be used for example, for detecting horizontal gene transfer, where you may have a piece of DNA that comes into a genome from a genome that has a different G plus C content, and that'll stick out right away in a G plus C content analysis. If we go up to 3-mers and look at codon usage in bacteria, they also have unique but highly conserved usage of different codons. This extends also to untranslated regions and may be primarily related to the GC content and the evolutionary relatedness of the organisms. So this heat map on the right shows in yellow the more frequently used codons, and in red the less frequently used codons for some bacterial genomes, which are in the columns, and for each codon, which is in the rows. So you can see that there's patterns here, and that they're related to the GC-rich or AT-richness of the genomes, which is in this upper column, or sorry, the upper row at the very top. You can see the block on the left is mostly consisting of AT-rich genomes, as is the block on the far right, and in the middle there's some GC-rich genomes, but it's not a perfect rule. But what you can see is that some codons are preferenced over others. So at the level of 3-mers, this is a useful analysis to look at how microbes differentiate themselves, and this can also be used for classification purposes. What another common Kmer length that's used is 4-mers, or sometimes 5-mers, especially for metagenomic binning. And you might have heard this referred to as tetranucleotide frequency. So this is essentially a conserved fingerprint that's used in the world of metagenomic binning tools to detect unique genomes by the patterns of 4-mers that they present. So in this figure on the right, this is a H. influenza genome, where each column is the 4-mer fingerprint of a 3,000 base pair region, so scanning across the whole genome. And each row is a unique Kmer, so there should be 4 to the 4, 256 rows here, each colored from low abundance in purple to high abundance in red. And a priori, if you didn't know that there was a conserved tetranucleotide pattern, you might expect this figure to look random, to have random splotches. Each gene, kind of each 3,000 base pair region, could have a different tetranucleotide frequency pattern, but it doesn't. In fact, it's quite conserved across the entire genome, except for a few specific regions. You'll notice a couple of them in the stars underneath the top panel of the figure here, where those indicate ribosomal RNA. And ribosomal RNA is so highly conserved over evolutionarily time that the pattern of the rest of the genome doesn't imprint itself as much on the ribosomal RNA as it does on other genes. Another divergence from the pattern you can see is on the far right, where the black bar indicates where a bacteriophage has integrated into this host genome. And it also has a very different tetranucleotide frequency. So when we talk later about metagenomic binning, we'll go into this in a bit more detail, But one of the things you have to be careful of is if you're using an algorithm to produce metagenomic assembled genomes that only uses tetranucleotide frequency, then essentially you're going to miss all of the horizontal gene transfer because it'll be over in another bin somewhere else. The same thing happens with 16S gene fragments. So this is an example of using this high dimensional data in a form that can be simplified to look at in 2 dimensions. So this is called dimensional reduction. And what it does is it takes the, in this case, 256 dimensional data that we get from breaking down the genome in the previous graph into Kmers and projecting it back into only 2 dimensions. So this is from a paper where they did a simulated study where they took a bunch of genomes, did this Kmer analysis, and then used TSNE, which is a machine learning algorithm, to re-embed all that data into 2 dimensions, and then colored it by which genome the contigs came from eventually, originally, and where each dot here is a contig or a small piece of the DNA. And you can see that, generally speaking, this technique separates out these different genomes quite nicely. The red is in one place, the blue is in another place, and there's some overlap between them, but it's not terribly extensive. If you look on the upper right, though, you'll see a little circle that says 16S RNA gene fragments. And that's, again, because the 16S RNA doesn't follow this tetranucleotide pattern like the rest of the genome. And so that's another thing to remember when you're doing metagenomic assembled genomes, is that you probably won't get the 16S gene in your bin. And that's a common problem with all of these techniques. We'll talk more about that later, though. Genome assembly also makes extensive use of Kmers using an algorithm called De Bruijn graphs. It's based on Kmer overlap, and we'll go into this in more detail when we go into the assembly. But I just wanted to show you quickly how Kmers are involved in this process. As we talked about earlier, we take a sequence and we break it down into all of its Kmers, and then you can assemble a graph of Kmers that are different by 1 base pair in a certain direction. So the first Kmer here is ATG GAAG. So we put that as the first node in the graph. And then we make a link to the next Kmer, which we can find here, TGG AAGT, and keep doing that until we come to the end of the sequence, where lo and behold, we find the same Kmer that we found originally. And what that means is that that goes back and links to the original Kmer. So the technique is very useful, but it's particularly useful when you get to longer and longer Kmers. If the Kmer length is too short, then many unrelated regions of the genome become assembled together. For example, we know from this red read on the left that the ATGGAAG Kmer is in 2 distinct places, but the algorithm can't know that because it's only using Kmers of length 7. So it would see those as identical and couldn't tell you which one comes first and which one comes after. It would form them into this circle. So on the right-hand side, I've shown a couple of assemblies of a Pseudomonas genome, where on the left, it's assembled with a Kmer length of 21 base pairs. And from that, you get 3,190 distinct contigs that are all in this spaghetti tangle mess here. But if you use a Kmer length of 127 base pairs, then you're reduced to 288 contigs. You can see, though, in the red regions, those are places where there's a lot of repeats that are longer than 127 base pairs. And so the assembler still can't find a way to resolve those repeats due to the Kmer length. So that's where long read technologies, which there's a discussion and a new channel opened up on the Slack this morning about long read technologies like Nanopore and PacBio. So if you're interested in those, check that out. but they become super helpful for genome assembly because they provide you much longer Kmers, and then you can do scaffolding and stuff like that. So to get on to the point of this lesson and the demo that we're going to do, Kmers are also extremely useful for taxonomic classification. And that's because as the Kmer length increases, the probability of a random match between two distantly related taxa decreases exponentially at the same rate that we saw the number of Kmers was growing earlier. So this can be used to classify DNA sequences based on their Kmer profiles by comparing the Kmers to a pre-computed database of taxonomically known Kmers. So what people do is they go to GenBank or to Silva or somewhere where you have a sequence and a known taxonomic identification. You make a database of all of the Kmers that belong to each unique taxonomic group. And then you go out and find some sequences from your environment of interest, break those into Kmers, and then compare those Kmers to the database and find out which ones overlap. A lot of the programs for taxonomic classification go another step further and recognize that not all Kmers are created equal. Some Kmers are more common than others, some are rarer than others, and they have different utility in determining the taxonomic resolution of an organism. So most of them will provide a tree, a phylogenetic or taxonomic tree, where the Kmers are mapped onto based on the lowest common ancestor of the organisms that contain that Kmer. As we'll see later, this technique is also used for identifying adapters and other contaminants in raw reads. For example, if you want to exclude reads in your metagenome that map to humans, for example, if you're studying seawater and you don't expect human reads to be there, then you can form a database and throw out any read that has a Kmer that matches any Kmer in humans. This is also used, we'll use it later in the demo, to identify Illumina adapters that end up in our final sequences and remove them. So this figure on the right shows an example of what I was saying before about how not all Kmers are created equal. In this case, this is the E. coli 16S position on the x-axis, and then the variability in the bases of a whole bunch of different soil ribosomal RNA genes that appear on the y-axis. And so a lot of the times when we're doing amplicon sequencing, we'll specifically choose to make our primers in the troughs and have them sequence across the hills so that we can identify the most variability in our sample as possible. So the V3, V4 region is very popular for that. But for doing taxonomic classification, it means that we could have problems where we find similar sequences or similar Kmers in two bacteria, for example, not because they are closely related, but because the 16S RNA gene is extremely conserved. So that's again why these taxonomic classifiers use as many Kmers as they can use, and they also have this lowest common ancestor algorithm to identify taxonomic specificity at different levels. So when you're choosing a metagenomic taxonomic classifier program, a few things you might want to keep in mind. One is which sequence format you're going to use, whether you're going to use nucleotides, where the Kmers grow at 4 to the K, or amino acid Kmers, where they grow at 20 to the K. May play a role in which one you choose. Most of the classifiers out there currently use nucleotides, and I only know of one that uses amino acids. Another thing you might want to look at is what database the classifier is classifying against, whether it's whole genomes or a set of marker genes or a smaller set of ribosomal RNA genes. And then you might want to look at which search algorithm it's also using. So we're talking primarily in this lesson about Kmers and Kmer indexing. But there's also other algorithms that are out there that don't necessarily use the Kmer paradigm. So the Burrows-Wheeler transform, BWT, will be talked about in a couple of lessons coming up, and is a very clever way to do sequence database comparisons that isn't based on the Kmer paradigm. Or you can use various BLAST-like algorithms which do fuzzy matching, which can be useful in its own way. Computers really like Kmers because they're exact and precise, and computers are lazy, so they would like to do exact matching whenever possible and not have any mismatches. So then you have to use slower algorithms like BLAST if you want that. Okay, so here's a big table from a review by Florian Breitweiser and Stephen Salzberg's group, where they compare a whole bunch of different metagenomic classification, and in this case, assembly algorithms. I'm just going to mention a couple of them here. I have not tried all of these. I've only tried a couple of them myself. And so there may be some discussion on the Slack group about what your favorite classifier is, and I think that would be interesting and useful. The ones I know that use Kmers and genomes that are quite common are the original Kraken, which I think some people are still using, probably because they went through the whole mess of building the database and are like terrified that they will have to do that again, and so they just keep using it. But don't use the original Kraken anymore, please. There's a new version, Kraken 2, which is much faster. The database building is much simpler and smaller, and you will be much happier not using the original Kraken anymore. Kraken 2 can also use amino acids instead of nucleotides, if you are so inclined. There's another version of Kraken Unique called Kraken Unique, which I forget what it does actually. We can look it up later. There's another program called Clark that uses Kmers for taxonomic classification. And then if you use Mother in the amplicon sequencing world, the classify seqs command also uses Kmers to compare against the 16S database. A couple of these other classifiers we can talk about later, but they don't use Kmers. They use the Burrows-Wheeler transform. For example, centrifuge is another one out of Stephen Salzberg's lab that uses nucleotides with this approach. And Kaiju is a nice web portal, actually, that will do this analysis using amino acids. So it converts all of your sequences into all 6 reading frames and then compares them to this their database. There's also a couple of packages that use the BWT with marker gene database. So MetaFlan and MetaFlan2, MOTU, and Amphora.net are some of those. And those may be discussed later, or maybe in the functional annotation side of things. So now we're going to head into the demo side of things, and we're going to go through an example of trimming some raw reads using a Kmer-based approach in bbtools, and a very simple taxonomic classification exercise using Kraken 2. And we don't really need to go into these, except to say, like I said before, that Kraken 2 is much faster, and the database builds much faster and is much smaller than previous iterations. So if you're using Kraken 1 and your database size was 240 gigabytes, The new one will be a much trimmer 34.7 gigabytes. And so there's a recent paper where I think they used Kraken to do 16S amplicon taxonomic classification, and made the claim that it was better than Chime, which I'm sure pisses off the Chime people. But you can try it out for what you would like to do with it. There's also, I wanted to mention a couple of extra and pre-built databases available for Kraken 2. So there's these 16S databases, Green Genes, RDP, and Silva. Please do not use Green Genes or RDP. Just use Silva. That's I shouldn't be stepping on amplicon sequencing people's toes, but that's my personal opinion. But for our case, there's also these databases maintained by Mike Lohman's lab, the Kraken 2 microbial databases and MaxiKraken. And then there's this genome taxonomy database, Kmer database that contains all of the genomes in that database, which is quite nice. So those are some other options you can use if you don't want to build the standard Kraken 2 database.